while my slides come up. I'm going to talk about the regulatory considerations for preclinical and clinical models. Many of you who have seen me talk before will see a lot of very similar slides. First of all, my disclaimer, if you're not used to seeing government employees talk, this is a pretty standard disclaimer. So what I'm going to talk about today, very high level, is what a biologics license application overview of what that looks like and what it takes to get a vaccine across the finish line. And then delve a little bit into licensure pathways, the traditional approval, accelerated approval, and animal rule approval. And you can see where dengue vaccines fit in. And then hopefully I've identified some areas where the researchers here and whoever is listening online might identify some places where you can help move this forward for everyone, including industry. Very high-level overview of what it takes to bring a vaccine to market. This is almost an offensive slide in some ways. This represents probably maybe 100 years of work in one slide. What the basic idea is that when you're working in the lab, you come up with a candidate vaccine, you test it in animals, et cetera. We don't regulate any place there. But when you have a candidate, it looks good, you want to go into people, that's when you come to the FDA with what's called an investigational new drug application, or an IND. And then you develop your product through phase 1, phase 2, phase 3. If all the data looks good, then you submit what's called a biologics licensing application, or BLA. We review that with you back and forth, and if the data looks good there, then you get a license and you can sell your vaccine or drug. And one of the other things I want to bring out of this slide, you can see in the bottom, safety, effectiveness, and manufacturing consistency. Throughout the development phase and through BLA and forward, we're always very, very concerned with safety, no matter what. If there's a safety signal, I'm telling you, it'll be a screech and everybody will get in a room and meet about it, no matter where you are in development. Effectiveness and manufacturing consistency are the type of thing that you develop as your product develops through phase 2, 3, et cetera. So in this process, I'm just going to focus on the BLA today and maybe what some of these animal models and clinical models can add to hopefully shortening the development and review phase of the BLA. So I always like to put this slide up in some way or shape or form in one of my talks, just to show the difference between what a review of an IND looks like versus a BLA. An IND is just a handful of reviewers, usually a regulatory project manager. That's where I got my start at the FDA. Clinical reviewer, product reviewer, tox reviewer, and sometimes a stats reviewer. When you get to the BLA phase, it increases in 2-3-fold amount of reviewers. And we really do go through everything with a fine-tooth comb to make sure that every product out there that you see has been really through the ringers, and you can be assured that it's safe and effective. So what is a BLA? A BLA is a marketing application. And the goal of a BLA is that a sponsor submits data so that we can ensure that a biologic product is safe, pure, and potent. A little bit more on that in a later slide. That the proposed benefits outweigh the risks. Labeling is adequate. I'm not going to go into labeling. That's a huge discussion and review issue. And then manufacturing consistency and the facilities where the vaccine is made are consistent and are up to snuff. So, like I said, we're charged at a very high level under the Public Health Service Act to ensure that a drug or vaccine is safe, pure, and potent. I put the citations there for the regulations. If you are having trouble sleeping tonight, go ahead and look those up. You go down, no problem. But I want to just go through these at a very high level. Safety is defined in the regs as the freedom from harmful effects to persons who are affected by the disease of interest. And importantly, at the end of that, it is the consideration takes into account the condition of the recipient at the time. So if you think about that, safety is a little bit subjective. Something like an anti-cancer drug for somebody who's in stage 4 cancer has a different safety consideration than a newborn who might be receiving a vaccine. And in the Office of Vaccines, we have a very low appetite for safety signals because generally we administer vaccines into a safe population and a very healthy population. Purity is what you would think. You say what's in the vaccine. You have to prove that what's in the vaccine is what you say is in there, and there's no extraneous materials. And then potency has long been defined as effectiveness. In other words, is your vaccine safe, and then is it effective? So in order to demonstrate safety, purity, and potency, we expect that trials will be adequate and well-controlled. What that means is an adequate and well-controlled studies, if there's a clear statement of the objectives and description of the methods of analysis, so it's got to be very well detailed what you're going to do and how you're going to do it. The protocol should be made and defined so that it permits a valid comparison to provide the assessment of vaccine effect. They are generally, we like to see placebo-controlled or some type of dose comparator or active control type of a design. So you compare your vaccine to either an active control or to a placebo. Sometimes, apparently, we have accepted historical controls, although I can tell you in all my time at the FDA, I've never seen a historical control. And then you have to be able to demonstrate the quality of your evidence. That's good manufacturing protocol, or I'm sorry, good clinical trial protocol. So phase 3 clinical trials that we usually see in the Office of Vaccines at FDA, they are typically double-blinded, randomized, controlled trials. There has to be a very good case definition, so that is what is a case in this conference of dengue, and that's usually worked out between the FDA and sponsors before they start their trial. For dengue, it's usually something like 2 days of fever, 100.5 or higher, and usually then you come in and you have a blood taken in PCR to confirm dengue. It's something like that. The trial size should be large enough so that it can, the power calculations can estimate effectiveness, which is usually a lower bound of the 95% confidence interval, exceeds some pre-specified level. Usually that's something like the lower bound of the 95% confidence interval is something like 25% or higher. Some cases 30, 40, 50%, and in some cases it's 0%. You can imagine if we had a TB vaccine, for example, that had some type of efficacy that was clinically or that was statistically significant with a lower bound of 1 or 2%, that would still be a great leap forward. So something like that would be an outlier, but important exception. So here is a very, very high-level overview of what a phase 3 clinical trial looks like for a dengue vaccine. And I'm going to come back to this slide at the end as my kind of, summary slide. But the basic idea is that you have some type of vaccine administration, 1, 2, 3 doses, and then at some point the sponsor will say, we have enough immunogenicity on board to start looking at efficacy. Maybe that's 7 days after the last dose, 14, maybe 28 days after the last dose. And then you start collecting efficacy and you watch people go away and come back to see, did you get dengue? And you usually do that for 1 year after, 28 days after the last dose, for example. And you collect efficacy for a year. And that measure after 1 year is your vaccine efficacy. Did you have, 50%? Did you have 80%, et cetera? But what's kind of unique and very difficult about a dengue study is then we continue to follow subjects for 5 years. It starts with sometimes it's called the hospitalization phase. It could be active or passive surveillance. And what you're really looking for is what we're looking for at the FDA and the sponsors are looking for is any type of a signal of vaccine-induced severe dengue, and any type of ADE or whatever you want to call it. And that is a long and difficult process. We acknowledge at the FDA this is a difficult and expensive trial. I know we have colleagues from industry in the audience. They can tell you how much this costs. I have no idea, but I wouldn't be surprised if it was an obscene number. And I'll get back to that slide at the end. So, licensure pathways. To get across the finish line with a BLA, there's 3 basic pathways that you can show that your vaccine is safe and effective. They're called a traditional approval. That's kind of what we just call it. That's not an official name, but traditional approval, accelerated approval, and animal rule approval. These 3 different pathways don't change in the sense that you always have got to show that your vaccine is safe, and you've got to show that you can consistently manufacture your vaccines. There's no difference between these 3 pathways in those sense. What's different is the way that you determine efficacy, or effectiveness, I should say. The way you determine effectiveness. And another thing I want to bring out in this slide is that the basic idea is that it kind of moves left to right. If you can conduct a trial to demonstrate approval through a traditional approval pathway, you need to do it through traditional approval. If that's not available, then you can look at accelerated approval. If accelerated approval is not available, then we can talk about animal rule. So a very high level of what is traditional approval. This is your standard clinical trial that you're used to seeing. The idea is that you give, for example, 10,000 people the dengue vaccine, 10,000 people get placebo, and then you watch to go to see who got a clinical disease. And hopefully everybody who got clinical confirmed dengue cases are in the placebo group and nobody in the vaccine. That would be a wonderful result. Sometimes you can use for traditional approval what's called a well-established immunologic marker that predicts protection. There is not one of those, there's no marker like that available for dengue at this time. An example I like to use for traditional approval with an immunologic marker is your Neisseria meningitidis vaccines. For example, there's others, but the human serum bactericidal assay has been used for a traditional approval for Neisseria meningitidis vaccines. Some considerations for a dengue vaccine. Like I said, there's no immunologic marker, so you need to do some type of clinical efficacy trial. It is very difficult, as everyone in this audience knows, to predict where and when dengue cases will show up, so it's a big challenge. Ideally, when you're designing a trial, we'd like to see prevention of disease in all 4 dengue serotypes. And importantly, and this keeps showing up, and this is what everybody in this audience knows, you need to demonstrate in your clinical trial that your vaccine is not inducing enhanced disease, as vaccine titer or as antibody titers wane. An example of traditional approval was Denvaxia. Denvaxia was approved based on the efficacy against virologically confirmed dengue on 2 randomized double-blind clinical efficacy studies. They were CYD14 and CYD15. However, we observed, and Sanofi observed, starting 1 year after vaccination, that they started seeing an imbalance of severe or hospitalized dengue, and therefore the indication was limited to individuals 9 through 16 years of age with laboratory confirmed previous dengue infection and living in endemic areas. That age indication was later expanded down to 6 years of age. On to accelerated approval. For accelerated approval, the way it works is, or the qualifying criteria is that your drug or vaccine needs to treat a serious condition, and it generally has a meaningful advantage over available therapies. Those 2 criteria actually fit for dengue. It is a serious, there's no doubt, dengue is a serious condition, and there's no real really good therapies, at least in the US, out there that are available. So you would qualify for those criteria. And the way it works is you demonstrate efficacy with what's called a surrogate endpoint that is reasonably likely to predict clinical benefit. And usually for a vaccine, that is some type of an immunogenicity marker, vaccine titer or antibody titer. And then after you get approval, you've got to demonstrate clinical efficacy in a post-licensure study. An example, I'll go through this very quickly because I'm running low on time. Prevnar 13 was approved with traditional approval based on immunogenicity using an OPA assay for the prevention of pneumonia or invasive pneumococcal disease. You can see it was based on these studies here. They were safety and immunogenicity studies, and it was later confirmed using what was called the CAPITA study, which was a very large, double-blind clinical efficacy study in almost 85,000 people, and they actually were able to confirm efficacy, and now they have full approval. Some considerations for accelerated approval for a dengue vaccine. There is currently circulating dengue, so you can conduct a clinical efficacy study, which means accelerated approval really isn't on the table. Additionally, there's no established surrogate marker likely to predict benefit or to predict clinical benefit. And then finally, even if we could go down this road, we would still need to demonstrate that the vaccine did not result in enhanced disease, which means you still have got to conduct that large study. Finally, the animal rule approval pathway, I'm going to blow through this because this doesn't really apply to dengue. The way this works is you demonstrate efficacy in an animal model. Actually, you need to do it in 2 animal models or more. And then if it looks good, you usually protect 2 animal models against challenge. That's usually the way it works. You have to conduct a post-licensure efficacy study as well to demonstrate effectiveness. The problem, or clinical efficacy, the problem with animal rule for dengue is there's not even really one good animal model for the purposes of animal rule that would demonstrate a clinical benefit. And also, like with all the other licensure pathways, you still need to demonstrate that your vaccine does not lead to enhanced disease. You'd still need to conduct that long study. A recent example of the animal rule approval, we just licensed Cyfendus last summer. This is a anthrax vaccine for the prevention of post-exposure post-exposure prophylaxis against suspected or confirmed Bacillus anthracis exposure. It was based on these studies below in guinea pigs and in non-human primates, as well as some safety and immunogenicity studies that were conducted in humans. So here's a very, very high-level overview of what I just told you. This is kind of a catch-all slide, basically showing that the major difference between these approval pathways is how you demonstrate vaccine efficacy. So a summary of what I've been talking about. The U.S. licensed vaccines requires data demonstrating that it is safe, a vaccine is safe, pure, and potent. Potency or effectiveness must be demonstrated in an adequate and well-controlled clinical trial measuring protection against disease. There's currently no well-established marker that we can use to shorten that time frame. And importantly, safety against the vaccine, against enhanced disease, must be demonstrated in an adequate and well-controlled clinical trial. And that's really the hardest part of a dengue trial. So in my 1 minute left, this is my final slide. I'm coming back to this. What I want to bring out about this is in many ways, among other impediments to a dengue vaccine, this is another impediment, this trial design and size and scope. It is hard for manufacturers to justify running this unless they have great data, because it's expensive, it's long, and frankly, I think it's a little scary because you don't know what's going to come up in those 5 years of if a severe dengue signal is going to come up. And what I would charge the audience is maybe a talking point to start a discussion is how can we shorten these 5 years? what can we demonstrate, maybe not a correlate of protection, but something of a correlate of safety, would be a huge, huge advance, if not to shorten the trial, maybe at least to weed out vaccines that would maybe fail later on in development. And I've run out of time, but that is something that I personally would love to see moving forward, because if you could get rid of those 5 years in some way, you would see a lot more people come to the table with vaccines. So that is it. Thank you very much.